All right, let's go ahead and get talking. I think we started to present some of this stuff earlier in January and it just didn't work out. But what I'm going to talk about first, best management practices and alternatives for managing fruit rots, other diseases of strawberry. This is the work done by myself and my lab manager, Mackenzie. We'll jump right in. Okay, first I'll talk a little bit about succeeding with biopesticides. They're becoming more popular. The various agencies are reducing our ability to use multi-site fungicides and other types of more traditional conventional fungicides. And a lot of consumers really want us to start using biorationals, biopesticides. So I'll talk about how to succeed with them, how to implement them, and then I'll show a bunch of the data, and then we'll move to fruit rots, which I think is another topic that people are interested in. What are these things? They're naturally derived pesticidal substance, they're plant extracts, they're minerals. In most cases, we think of them as microorganisms that have been forced to make some sort of product, a secondary metabolite. And interestingly enough, the EPA even includes transgenes as a type of biological produced pesticide. Now, can we use those? No, but they're technically part of the group. They vary in type, cost, efficacy. The nice thing about them is they have multiple modes of action in many instances, and low risk for resistance. You can find this definition if you were to click on this link in there. And then this is the EPA's list, and maybe we'll click over just very briefly and take a peek if it shows up. Ah, there it is. And yeah, if you were to look on that, you could see there. Well, now it's gone. There's no alt tab in Macintosh. Oh, there it is. Great. Yeah. Tells you what they are, same definitions I gave, and you can find the list of the different products as well. There's the actives right there. You click on that link. The other thing that there is the FDA has a series of antimicrobials that are part of food grade things, and these are called GRAS, generally regarded as safe. They can be extracts in food and other stuff as well. And they're often a part of our pantheon of tools. And then finally, we got EPA minimum risk pesticides, and they have their own, every one of these things has their own little link. You can kind of click on them and read about what the different things they are. There's the notice inventory, which means these are generally very safe. And then of course, 25B, do we still have the page? Now it looks like it has changed a little bit. I wouldn't worry about it. Let's try the top one. Yeah, no, it's not anymore. Oh, that one works. But anyway, not too much to worry about. How do they work? There's a bunch of different diseases. The same talk applies whether you're doing apples or strawberries or grapes. They all work the same. Modes of action include they either make some kind of antibiotic metabolite that kills the microbes when they're present on the plant part. They are forced to produce this in a soup. Second option is that they can kind of compete for plant tissue on the surfaces of flowers. That usually works best with this one, this niche competition. Kind of hard otherwise to do sort of keep these things off the leaves and the fruit. When you go for competitive inhibition, you want flowers. And then the final method is sort of defense inducers. They're going to sort of slow invasion infection by microbes by trying to boost up natural plant defenses. And they work not so great compared to a real serious fungicide, but they can do some stuff. And we'll see them at work as well. For the most part, these things are protectants and eradicants. They only really protect against new infections or eradicate pathogens on the surface. And they're very limited in how long they're going to last. They're not going to last forever. And you might have seen some of these things, double nickel, Serenade, Serafel, Tegro. A lot of these are a lot of microbial soup products. This is a yeast fracture. This is a protein from a plant called the lupin, polyoxin D. This is sort of more closely related to an actual antibiotic, but it's naturally produced. And these things in green are your various grass type products, food grade essential oils like thyme, cinnamon, and other stuff like that. And then finally, what do we have in the defense inducer world? These will sort of work internally to turn on plant defenses, natural defenses. Actigard is a synthetic one. Can't really use that in organic, but it's still doing the same type of thing. LifeGuard is a bacterium that will turn on defenses. This is an extract from a giant knotweed that'll damage the plants so much that I think they're being attacked. This is another classic defense inducer, as well as Vasaplant. And then even things like plant growth regulators, PGRs, which aren't really used much in the small fruit world unless you're illegally applying apogee for runner control. But these have various other defense properties in many crops as well. Why would you want to do it? Consumer appeal and acceptance. People really like these things. If you want to get into the organic markets, that's my organic apple orchard. You can always tell an organic apple orchard by the fact it doesn't have treated wood in it. Like that's your sort of first clue. The second clue is it looks terrible in terms of yield and disease when you get up close. If you have covered operation, if you have tunnels, and this is where strawberries really shine in the berry products because you can do them under sort of covered environment agriculture. If your operation is small and frequent spraying is possible, you can make these things work. And then finally, disease pressure is light. And then the right types of pathogen. This is powdery mildew on apple. If you get it on your small fruit, you see the spores are just right there and present. We'll switch up to the old laser pointer. Seems to be a little bit better right there. Oh, or if it's fire blight, there's ooze right there. These antimicrobials can just jump on and get them. So how do you do best with it? The first thing you do is you start with your horticultural practices. And you can imagine this the same type of thing whether you're going raspberries, blueberries, or apples in this case. If you have a well-spaced planting, excellent horticulture, you're going to find your fruit is better yield per acre. You're doing all the best horticultural techniques. You're going to have easier disease control. You take a look at these apple orchards that we have, a more traditional orchard from a more pick your own area. Trees are packed together. They're big. They're bushy. It's not a good use of land in terms of fruit per acre. And then you have a super spindle. You can see immediately, oh, the air is going to circulate much better through this planting. Keep your weeds out. Keep everything out. Air will be one of your best materials for managing disease. You can see that through the planting. Great air circulation in that particular block. Pretty terrible there. If you were to go into these different blocks, you'd see one of them's loaded with disease, the other not. And that's with doing nothing. Of course, when you start making applications, you're in better shape. The second thing we like to promote is the use of forecasting tools. We'll have one for strawberries. I'll show it a little bit as well. They'll predict the conditions that sort of will predict the pathogen biology and maturity, pathogen dispersal, and potential for infection based on weather events that are happening. It's becoming a little more challenging with our weird erratic weather, whether or not it's 40 today and -7 tomorrow, or whether or not it's 40 today and 70 tomorrow. Sometimes these things kind of cause the models to behave a little strangely. The pathogens can't really correct on the dime for these erratic changes, so it might give you a little bit of more a warning that could be a little more erroneous than usual. If you do these, and I think we'll see some data in a few minutes on strawberries, you'll see that you'll be able to increase the precision and effectiveness of whatever management tool you're using, whether it's conventional fungicides or biopesticides over a rather calendar schedule. And the erratic weather can be a little bit tricky, but if you're going on a calendar, you're definitely going to feel the pressure of bizarre rain and temperature fluctuations and whatnot. We have this one for NUA. I think it is open in your area, at least up in for several places up in there. And if you're not part of the NUA network, your growers or your organization can join on your behalf, and we'll open up weather sites all around your state, and you'll be able to use some of the same tools I'm showing. This one's for apple scab. We don't really need to pay attention to that. But it's nice about it, you can have your own local weather station that will feed directly into it and be perfect for your farm. But even if you don't have that, you can use a nearby leaf wetness and temperature algorithms from airports and other sites in the region. And there are a good many of those that are brought into the network as well. So how to do these? So we started a series of 6 years of trials. And it first started off with like, well, could we use biopesticides, period? And then the second one was there's a lot of regulatory concern about Captan. And we thought, well, and we went straight to Canada. They were interested in this type of work. And they're like, whoa, what about Captan? And so then we started on another 3-year series of studies on how we could use just biologicals plus classic single-site fungicides, the good products in place of, and just remove away from Captan, which was under regulatory heat. And in our trials, we'll be looking at all kinds of things, gray mold, anthracnose, the common leaf spot, and leaf blights as well. And then we've already talked a lot about there's a huge market interest in fruit that were produced without multi-site fungicides, no mancozeb, no copper, no Captan, and we want them with biopesticides. We've done a lot of work in apple, and a lot of concern with getting away from Captan and stuff is the idea that you might develop fungicide resistance to some of the really fancy single-site fungicides, like a Maravon or a switch or something really nice like that. Those have the potential to develop resistance. But the idea being is maybe we could use a biopesticides multi-site mode of action to make these sort of resistance changes and sort of guard against that. So we did a bunch of studies. We did 3 prior. We're doing 3 now. We like these double row, low tunnel planting, 12 inches apart. We did this later in the season with day neutral strawberries. And we have 4 replicate plots at 30 plants. We do a bunch of assessments. And in this, one of the first factors we'll be looking at is the strawberry disease model that's present in NUA. It will predict for botrytis and anthracnose, and kind of see my team setting up these little plots there. And it's a fairly complicated statistic design, but in general, you have 3 levels. The first one is whether or not the strawberries have cover on them. Are they under a plot, or are they wide open to the environment? And then second, within each one of those big plots, we ask the question, what if I time weekly on a calendar schedule or versus using the NUA model? And if the infection risk is below this 0.4 threshold, we don't do anything at all. We just let it happen and see what happens. And then within that, we either do an integrated biopesticide program if the infection risk is between 0.4 and 0.75. We use double nickel alternated with LifeGuard. This is defense inducer. This is antimicrobial. And if it's greater than that, we'll go switch your Maravon, the big gold standard heavy hitter materials. And comparison to that, if we same things, instead of this case, we'll go Captan instead of a biopesticide, or we'll use one of the fancy products. So we'll show a couple years of data. We started this in 2021, sort of our application timing. We start the experiment late because my job is really apples, but we are interested in berries as well. So we're running it for about 8, or I guess it looks like 9 weeks in this particular thing, and going every week with a calendar application. If we use just timing by the models, you only get 6 applications needed, and they're in pink. And you can see these are the risk levels. They're quite high during this pretty wet year. And because they were so high, our little protocol called for 5 single-site applications, or 1 biopesticide or 1 Captan application as well. If you go on a calendar, you're applying all the time. So 9 versus 6. 2022 is a little different year. We ran it for about 10 weeks. It was moderately raining, a lot of late season storms. 21 was pretty hefty. You can see it starts right out of the bat there with some high levels of gray mold and anthracnose. It predicted 10 applications, 5 NUA, 3 single site, and 2 biopesticide applications right there. You can see immediately that it's not calling for as many applications in the season, with a rainy period happening at the beginning and end of the experiment. So you really don't need to go in that much. And then 23, pretty dry and cold. We're only using 1 switch Maravon at all. And we ran this one out for about 8 weeks. Only 3 things were flagged by NUA, and it said that either biopesticide or Captan was fine. Not we didn't do anything, or we did biopesticide or Captan on the calendar schedule. So that's what it called for, but let's take a look at the data and see if we can see some different trends. One of the diseases that we look at is common leaf spot. It's that thing that makes the little white lesions on your strawberry plants with a red halo. We found out that just in general, this is how the graph looks. This is the incidence. This is how many leaves had common leaf spot. So about 40% would be our total. And this is if the plots were under cover, a tunnel, or completely white out in the open. The first thing that you can kind of see is it's a pretty hefty year. About 20% is a threshold that, if reached, would sort of compromise your ability of your strawberry to be productive. So in this year, everything was pretty much under the threshold, but you could see immediately the magic of having cover over your crop is a lot better. You end up, the bars tend to be a lot lower when you're under a tunnel than in the environment, particularly with this one particular year, which was going every week with Captan, was actually worse than using a biopesticide, which would be your little pink bars. The other thing you can see is that like NUA and the calendar are the same, but we're spraying a lot less with NUA. 2022, different year, you can really see the impact that tunnels can have in keeping that incidence low. And it doesn't matter, the incidence between the NUA plots and the calendar plots is about the same. When we went things with Captan, sometimes this dark pink bar seems to be a little higher than using a biopesticide. We'll talk about why that might be. 2023, it's a pretty hefty year. You can see immediately, man, having things under cover is great. We have a lot of failures, and particularly in the programs where we use Captan a lot, calendar and being in the uncovered. Even using the model, you still had a lot of trouble out in the open. And then the bottom line is like, yep, you get fairly similar between with our calendar schedule and NUA, but you're spraying a lot less. And we didn't really find an appreciable advantage using Captan over double nickel and the other stuff. What does it look like for the other diseases? Very similar patterns. We won't spend a lot of time looking at them all, but this is anthracnose and that sort of purple color, same type of trends. Tunnel is good. Using forecasting will save you a lot of applications and you get similar control. We're not seeing a lot of benefit from Captan. Same types of things here. Though this was an interesting one. Tunnels were less helpful because they were getting blown over all the time in the windstorms. So in that case, they didn't seem to do much for our fruit rods. That was a little different. In 2023, we got better at putting up the tunnels. It takes a little bit of practice learning how to implement these things, and we've been doing it for 6 years, and we're finally getting our handle on low tunnels. And now we've gone into the greenhouse, but we're really not seeing a big advantage for Captan. Anything with a darker bar has a lot of Captan use, and particularly in the uncovered. It's not. So you could see that using disease forecasting in tunnels is going to do pretty good for you. Same trends with gray mold. It's going to mirror the anthracnose. And of course, in that year when the tunnels were getting knocked down, it was just wet everywhere. It was kind of a disaster. But in 2023, you can really see those patterns. Being out in the open is terrible for gray mold, but being uncovered is great. And you just get almost identical stuff between whether you did forecasting or sprayed every week. And in many instances, not having Captan was better. And it's kind of interesting about why that might be. We have a lot of tunnels, and the rainy and erratic weather, tunnels can be less effective, like in that 2022, and you can get a lot of humidity locked in there. It'd be kind of trouble. In wet years, the models may call for more applications, but they're generally tend to be less than what you would do on a calendar schedule almost every time. And it seems like if you want to make these things work, use them with a model and alternate them with the real materials. When that threshold is high, then you go with Maravon, you go with Switch, you go with the good stuff on your fruit rots. And particularly in wet years, it's fine. And when it's dry, you do nothing, or you put out one of these biological products. But one thing that we noticed in our tunnels is that Captan residues were staying on along. And in the plant world, Captan is kind of phytotoxic, if you will, and has really slow drying conditions. If this happens in apples, you'll get a lot of injury. And we were wondering if this was happening to a little bit underneath the tunnels, and the Captan was just not drying off enough. Enough humidity was in there and causing more injury to the tissues, predisposing them for leaf spots and fruit rots. Now, maybe in an open field it would have been better, but man, those Captan residues were staying on for quite some time. And maybe in our plot, it wasn't as effective as we would have liked it to be. Let's see, we're about 7:23. I want to make sure we talk about fruit rots. Let's see if we have any other interesting things. Same type of setups and biopesticide trials. Some new products that are coming to the market. Common leaf spot. Again, this is like, oh, how many either leaf spot on the leaves on strawberry plants? What's the percentage? Untreated blots could get up about 40% of all the leaves covered. You can imagine a bar about 20. Being a good one. Some newer things that are coming on, there's Lifeguard by itself. Even though it's a very soft biopesticide product that induces resistance, not too bad. We had a really nice time with Solmatrix, which is like potassium silicate, and in theory should be taken up by your plants and strengthen the tissues. In this case, we found that it was quite nice against leaf spot. And then there's a couple new products that are these low-rate copper products, really low levels of copper. They get enhanced by zinc. And they're coming out probably this year or next in the Curzin and Securzin group. And they will do both fungi and bacteria. So we were fairly happy to see those. What else did we do in these? Some more life garden cures. Very similar things. Anthracnose can get very high in some of our plots, up to 60% of fruit rot in many years. Same sort of trends. We really are liking that cell matrix pretty good, and some of these other products from these cures and things being sort of softer products and low input coppers are doing pretty strong. Gray mold, that same year, they're all kind of very similar across the board. But you can do pretty well. It's been a pretty rainy, cold season. A lot of botrytis and these different products, even the defense inducers and stuff like Matrix, were able to do pretty well. What do we say about this? Biopests, they're pretty effective if you use them with single-site fungicides overall. We were very excited to also see that Matrix, a silicon product, been fairly consistently effective. It can either structurally fortify, or if you think about using silicon lubricants, you can imagine spores and stuff sliding off leaves and fruit and falling on the ground instead of sticking and infecting. Kind of interesting stuff. And these are sort of low MCE copper products that get enhanced by putting zinc. And a little bit of zinc makes the copper work well and puts less copper in the environment. These are very easy to mix and apply. And we've done a lot of studies looking at these with adjuvants, and they seem to be pretty good. So at this point, I'll take any questions you might have about this stuff, and we'll switch up to fruit rots in the meantime. I'll put this one away. If there are no questions, we can go right into fruit rots too. I'll look in the chat. Just type something in there. It looks like we're pretty good to go. We can also do a few more at the end, but this next talk is a little frustrating because soil-borne diseases are hitting New York pretty heavily, and they can be very frustrating to identify and manage. And we'll talk about some of the tips and things that we've used for dealing with the most common ones. One of the best, the most important things that everyone wants to know in New York is how to identify these things and how do we manage them. They can be really challenging to identify. In many instances, you're just going to get a physiological symptom that results from a pathogen infection. One of the more common ones is you might just see yellowing of the foliage, such as in these beautiful blue raspberries that are infected with a fusarium or a verticillium type issue. Just some chlorosis. They might just start wilting, or they could end up having full-on necrosis. And a lot of these things are general physiological reactions and can be due to any number of problems and not just necessarily root disease, particularly wilt, necrosis. If you get too much water, they'll rot without a pathogen. If you don't get enough, they'll wilt on their own. If you don't get the nutrition right, they'll look like that yellow raspberry as well. So how do you even begin to parse these sort of things out? The first place is you look at the distribution of symptoms. Patchy versus uniform over the planting. Now, if it's a disease, it will probably be in spots. If we go back for a brief minute and look at this raspberry here, switching up to the laser pointer, you can see that the other plants around it are probably green. If you see it localized in a spot, you might think, okay, it's my first indication that this is disease. Now, if it's the one edge of the entire planting, it might be abiotic. The other thing that you can do to sort of weed out abiotic is if you see the symptoms on other plants that aren't the host or the thing you're planting. If you have strawberries and you notice all the weeds look to be affected, you may be like, wait a minute, that's abiotic. The diseases are very host specific compared to the other types of abiotic injuries. So in these cases, soil-borne diseases, you're going to see them sort of clustered in areas where the pathogen population is happy. The pathogens love wet spots. So if you see something that looks like it could be a disease and the area is wet and it's all isolated in a wet part of the field, you think, yep, probably disease. If it's so dry you have to mine the field with a pickaxe in order to like harvest your crop, you think, wait, this is probably drought or abiotic injury. And they can be a little tricky with winter injury, which we're all sort of dealing with all the time, particularly in the spring. The other thing that will happen is you'll see the diseases, the symptoms will keep developing over time and spread. If it doesn't spread, it's probably something else. Now, if you just see a few symptomatic plants and you don't panic yet, but when the whole planting starts to look symptomatic or is progressively becoming symptomatic, that's when you need to sort of take action. A couple of different things that you might see. This is actually some winter injury in raspberries. The buds were broken on the floricanes, and then they sort of just look like this sort of hazy fog. Well, what is that? This is another winter injury as well. You might notice the straw cover is gone. It's kind of patchy, but it looks very, very dry. And you're like, oh. But in this case, they need to panic, but it's most likely due to a loss of winter injury. And then, of course, once you've already got the planting, everything that you want to do to manage diseases happens prior to the planting. When you saw all the stuff, we were looking at forecast models and treating disease. You don't see it until it's almost too late. You can't do anything. And then, of course, you don't even see the root diseases until you've already got the plants in the ground. Makes it really, really frustrating. Anything you do after the fact, not really effective. It just slows down the spread, and you can't cure the infected plants. The other thing that can happen with root diseases is that if you get one and it looks like it's starting to die or mostly dead, they're really frustrating to have because they're all in the soil. You can't see them. They're hidden. And all the diagnostic symptoms are below ground. So if you wanted to look at the roots, you have to kill a plant or look at it early, and then you're just losing some of your crop as you pull them up to check on them every now and then. The other thing that happens is once the plant starts to die, it just becomes this fungal decay free for all, like a winter injury or anything. Once it starts to die, the original pathogen just gets overwhelmed and other junk fungi come in there and cause trouble. So how do we usually do it? In the case of strawberries, I like to cut through the crown of the dying plant. This is sort of, as you imagine, sort of a bare root plant, and you'll just want to cut down the length of it into the different roots. If you see inside, if it's injury, the cortex of the crown will become brown and the vascular tissue is sort of white. This would be more of a winter injury, and it can be localized right there if the middle kind of freezes. If the vascular tissue looks white, as in here, and the rest of the strawberry looks brown and necrotic, it's probably just a winter injury. All the pathogens want to eat the vascular tissue. That's where all the nutrients are, and in many instances. And so this is verticillium. Sometimes it can have a bluish cast. Healthy, if everything is white, you have a completely different problem. If you got Phytophthora, it'll be red, particularly in the vascular tissue, and it will also spread to some of the outer sections of the plant. Black root rot, very similar to Phytophthora. In this case, it will look kind of like winter injury, except it'll be darker and more patchy. That one's a little trickier to diagnose. The management practices are kind of the same for both, sadly enough. Here's an example. I've cut the other way across the crown. You really want to go right down there, but you can see the core in the middle is completely browned out. And this case, this plug was frozen in the very center, and then when it warmed up, it just died. But the vascular tissue you can see there and there is clean and good. And if you were to strip down the length of the roots with a pocket knife and you look underneath the black epidermal covering and it looks white, then you know all is well in the world. So some of the things that can happen, and this will happen sometimes in the spring and March that's coming up, if you were to have plants overwintering, and the straw blows off here. In this case, you could kind of see, oh, the straw, this is a rocky, really rocky soil, and the straw has all blown into the middles of the rows, leaving the plants kind of exposed. It doesn't look particularly wet, and and depending on the time of year, sort of a classic spring freeze out, death looking thing. The other thing that can happen as well is if you have a lot of disease, like even leaf spot, and you go into winter, you don't have to have a hard freeze to kill it. Any disease from the previous year can predispose your plants to winter injury, even if you're doing everything right. They'll just have reduced vigor, look unthrifty, even dead. Again, this is the example we've already seen. Cortex brown in the middle, white vascular tissue looking really healthy. In this case, this is again in raspberries. You can kind of see this like, whoa, why is this haze of looking dead stuff? In this case, this was winter injury in raspberries. Temperature warms, floricanes that will be producing flowers and fruit appear to wilt and die, but the primocanes are hardier, like you can see down there near the bottom, and are a little more healthy. But the stuff that you want looks kind of wilted and dead, which is kind of frustrating. And let's see. And then, of course, the other thing that'll happen is the injury can be most severe at the growing tips. In that case, you can begin to think winter injury. It's rare that canes can be killed to the ground, but if you have a really sensitive variety, this can happen. Buds primarily get affected. And then sometimes in strawberry, if you get a winter injury, it'll do fasciation, which will look like it's gotten a really wide stem. It's like a whole bunch of runners and stuff came together like and they're glued. They look kind of mutant. That is a meristem injury that caused the plant to flatten. And it's caused by usually typically a winter injury. The thing that'll happen, buds will die and the winter injury plants will set up new primocanes and you're fine. But if it's a root disease, the entire plant will slowly die throughout the season. That's how you can kind of pick those out. Drought injury, not well irrigated, summer drought. A lot of things that can happen in this case is it will have a drought period, and then you try to compensate by overwatering, and then you water onto a hard pan, and that water pools at the crown and then rots it from a disease while it's suffering from injury. Just lack of water, the plant will wilt and shrivel. At this time as well, they'll become susceptible to chemical injury. If you've never seen herbicide injury, but it's under drought conditions, it's more likely to happen. You might think, what on earth? If you remove the plant cut to the crown and it's hard and dry, like clay, you'll ask, has there been rainfall for 2 weeks? If they're fine roots, are they sinewy? Are they dry? Here's a raspberry here. Someone thought I had a root disease. You see the soil is kind of hard. Everything is white and clean on the inside, but it was really dry, really sinewy, really, really hard. And it was kind of like whittling more so than like cutting into a raspberry plant. And I was like, yeah, they probably had drought. And it was a drought season as well. But they were really worried that they might have overwatered. But you can see all those little roots are very white, just letting you know that's something else. Verticillium wilt, this is a troublemaker. It can be on many hosts, including weeds and vegetables. If you have a diverse farm, you can get verticillium and other things, and it'll come into your strawberries. And this one's somewhat diagnostic. The above ground symptoms are fairly distinct. The outer leaves will die first, as you can see in this picture up there, or you can start to see down here on the bottom of this strawberry plant. They're in a very weedy looking planting. They're starting to die on the outside, and the middles look good in the middle. And the younger leaves will eventually become a little stunted. You can kind of see they're not, this one's not suffering as bad as some of these ones in the picture. Little islands of green and death. If you were to bring it up and slice it through the middle, you can see severish bluish streaks on the runners and the petioles and in the vascular tissue. Yeah, we've already got a late rot happening here. It's typically after the decline, but you can see the verticillium there, and it's right there in that vascular bundle, and making the plants look fairly blue looking, and they can have kind of a bluish cast. It's not going to be Kool-Aid blue, but it will be blue enough. Same thing can kind of happen in black raspberries. If you own any of these things, they're fairly sensitive, the varieties are to verticillium. Well, it'll start, and that's what the picture was earlier. It wasn't drought or anything else. It's this is a verticillium wilt from a horticulture breeding block. And the youngest canes wilt first, petioles will remain attached, and the youngest leaves will be green at the bottom, but the tips will be curled over, much like you're seeing there. And they will get a very bluish cast, and this is more advanced stage. And then sometimes you zoom really in, you can begin to see a little more bluish in the crux and stuff. But a blue raspberry is already going to turn blue when it dries anyway. How do you beat this thing? It can survive for years, unfortunately. Wait 3 years if you plant any of these things. Eggplants, peppers, if you have pigweed, if you got nettle, stone fruit trees. Look for replanting stock. There's no resistance. Switch to red raspberries. They get the Phytophthora. They don't get the verticillium as much. There's nothing to really do about it, which is kind of very sad. Fumigation is impractical nowadays. Drenches. We do have some stuff that we'd like to recommend. The what's the biofumigation and anaerobic soil disinfestation is really variable on how that works, and it's a lot of work for the farmer, and we haven't really seen a strong benefit from it. That's in another talk further down the line. Crown gall, usually a raspberry only. This is a bacterium that happens when you're doing work or renovating, infects the crown of the plant and causes trouble. They get a lot of stunting and wilt in the outer leaves, and then they can make winter injury bad. If you ever see it, you'll see these like tumor-like growths. if you ever get stock that has tumor-like growths on it, the raspberry plant is not a nitrogen-fixing organism. If it has galls, throw it out and ask for new ones. What'll end up happening, this is a really old gall here, is these galls will disintegrate in the winter and release bacteria. They can live in your soil and cause trouble. This is real common if you go in and plant after stone fruit or grapes. A really big problem in grapes. Never plant with any weird galls or growths. Just leave it out and then take care when you plant after these. And then watch out when renovating. Anything that damages the, weed eating or any of that stuff will do it. Insects can also sufficiently wound the soil line. In terms of some of the big ones, this Phytophthora root rot is a big problem. Everything. So anything I talk about here will apply to vegetables or any other crops that gets Phytophthora. This one's not a true fungus. What it is an aquatic organism that would just as well live in your irrigation pond as it would in your field. It likes wet soils, low-lying areas, and heavy rains. Consequences, it'll initially cause the plants to look stunted and chlorotic. You'll start to see maybe patches first where it's declining. This is where you want to focus your efforts on diagnosis. If it's already dead, don't even bother looking at it. And what'll happen is it'll be killing early. It likes cold weather. So it'll be killing in March and April when things are looking good, or right after you plant if you have a cold early season. And you'll start to see the outer leaves look, the middles look stunted. The outer leaves will burn a little bit, becoming blackened or brown on the outside. And then as the soil warms up, the roots are already rotten. And so the plant's like, I'm ready to take up water. And it can't because it doesn't have any roots. And then it just pretty much dies. Here's some examples of what this is a black root rot, very similar to Phytophthora, of strawberry. In this particular instance, it's a soil-borne compilation of nematodes, compaction in wet soils, and pythium, which is very similar. And it can be a little bit worse. You can kind of see it'll have a little bit of areas are mostly white, but they'll be blackened in spots, such as right there, or blackened right in there, but white elsewhere. This sort of patchiness gives you an indication. And this will happen over time. It's really gradually occurring if you have the plants in the field for multiple years. Cold injury, herbicides, compaction. Herbicides don't have to kill the strawberry. They just have to weaken it. Excessive water, a lot of work in there, people stomping, tractors and stuff moving. And as I mentioned before, it's pythium. It's kind of like Phytophthora. You can kind of see that some of these roots are white. And then here's white and then a black area right through there. Oh, it's kind of white and turning black through there. That sort of patchy splotchiness happens. It can be very endemic, and it just wrecks fields in New York. And typically you'll see it in the establishment years. There's another shot. I've got one right there. It kind of looks good. I mean, all the feeder roots are gone. That's your first indication that there's a serious problem. Like, oh, look pretty good. Oh, wait. It's turning blighted and black, and you can see these really, really dark areas. Vigor, stunting, death of plants. How do you beat a lot of these things? Once the plant's infected, you got to get it out. And the bad news is all the propagules are in the dead tissue, and they'll last for years in the soil. Phytophthora, black root rot, crown gall, verticillium, they make survival structures. They'll just be there forever. The best thing you can do is good drainage. Select a good site. Don't plant in a low-lying area. Always check your plant material. If you get 3,000 plants, check every couple hundred, cut it open, make sure it's white on the inside and has a healthy root system. Try to keep traffic out of your planting. Use raised beds if you can, and careful irrigation. If you have a drought and you might over irrigate, you can end up causing death as well. What else do we got? We've got existing plantings. Insulation during winter and spring freezes is essential. And then start to scout in the spring and early summer for the first signs of decline. If you see wilting beginning, you might be able to mitigate the loss. What can you use? Chemical control, Profite and Fostrol and Rampart. These are safe biopesticides. These work really well on Phytophthora and black root rot. They are a phosphorus acid materials. It's a phosphorus acid salt, and it will get translocated up and down the plant, and which is great because you want it to go straight to the roots for these root troublemakers. And how do you use this? I usually you have to wait till the green tissue is out because they are translocated. So you apply Profite Frost Tolerant Rampart to the foliage, and once every 30 days, and it gets translocated. Be careful if you mix these things with oils or ECs, SCs. ECs are usually worse than SCs, or Captan. I've injured a lot of crops with Captan and phosphorus acid fungicides. Really good. You can use mefenoxam or Phytophthora, but it costs more than gold. I always love the fact that they call it Ridamil Gold, because they might as well just call it gold, because that's how much it costs per acre to spray. You might as well be spraying gold at that point. You can do a fall soil application. There's resistance risk here. There's not resistance risk to these things. These were used recently in more recent years from airplanes in California to stop sudden oak death. So they're pretty good material. This is more on mefenoxam. It targets RNA polymerase. The fake fungus, Phytophthora and Pythium, can't make proteins and dyes. Highly resistant, but it's a single site, so it goes for mode of action. Here's a beautiful picture of a strawberry that looks like I dumped Kool-Aid in it. I cut this fresh, and it was just loaded with that red steel right in its vascular area, right in a little bit of the cortex. It's real scary stuff. Foliar drench for anything. The Pythium and the Phytophthora and a bunch of other things that attack golf courses are called heteroconts. They make a motile swimming spore. I mean, this stuff works great as long as you don't have resistance. Really good for black root rot, but the problem is in our regions over the years, people have kind of burned them out. Again, these are your phosphorus acid products. These are really good. I tend to recommend these. More pictures have some beautiful red steel right in there. Phostrol, they're labeled for these. They can be used in a greenhouse, so that's not bad. You can dip the plant into them. That's fantastic, but typically they need to take the material up and translocate it around. You can even put it in an irrigation line. Very success stories like in sudden oak death in California, happy growers, and sometimes they can even get real fungi as well, but it's not so tricky. What else can you do? I've seen some pretty good work with this product as well. Actinovate AG. It makes antibiotic metabolites. It's a microorganism that makes the soup that you put on the plant. Foliar and soil-borne. It'll do Phytophthora. It'll do regular fungi. If you get Rhizoc or something strange like that in your vegetables, you make a drench or a dip. Do it right before planting. I would definitely say you get a lot of bang for your buck with drenches and dips of this stuff, because you don't waste the material. You just dip it and plant really quickly, or you can have it soaking in it for just a little bit. Maybe you don't want your leaves with it. On that, pretty good control. If you ever have mummyberry, this is supposedly a pretty good one for mummyberry and does pretty well for soil-borne diseases. Classic material that we can end with, or we can continue to talk about other stuff if you want. Trichoderma harzianum root shield. You dip in there. We don't really have charcoal rot, but it looks like I might have found a couple instances in New York State. This fungus is just a terrible competitor. It'll outcompete everything. The bread mold in your fridge. It'll outcompete bathtub mold, and it'll outcompete root disease pathogens on strawberry. And once again, you dip and drench. Great on fungal. Great on those Phytophthora oomycete diseases. A lot of people have good luck with charcoal rot on strawberry, put it out in abandoned. History of successful application. And with that, we can take some questions, or I can talk about some other stuff as well. Sort of my members, this is sort of some of our field experimental plots. If you were going to do this, I would definitely recommend make a big row of those. But that's a real inexpensive way to have successful stuff. So we can take some questions.